make sure that others are joining. Can anyone tell me what did you learned yesterday from yesterday topic? If you're able to unmute, you can, but in case if you feel that it's not possible, you can go ahead typing in the chat box. I'm watching it.
We learned about various databases which are useful
Yeah.
in retrieving sequences of nucleotides and amino acids.
Okay, fine. Others, can you list out the databases which you have come across, or in case if you feel that we have missed something and you can add it, you can just type in your chat box. You're not able to access the chat box?
We
Yes,
went to
no.
databases such as NCBI, Uniprot,
Yes, very good.
EBL,
Okay,
DDBJ, and etc.
okay, very good. Yeah, I'm getting responses in the chat. You can speak to me in the chat because I'll continuously try to watch. That makes our communication easier. Okay. EMBL, European Molecular Biology Laboratory, right? EMBL or EBI. Okay. What is a sequence actually? What do we mean by the word sequence? It can be a nucleotide sequence or it can be an amino acid sequence, right? So
Yes, ma'am.
what are the nucleotide sequences? What are the 4 bases?
AGCT,
Adenine, guanine, cytosine,
thymine.
Thymine, fine.
AGCT.
Amino acids, how many standard amino acids are there?
20 standard
20.
amino acids.
20 standard amino acids are there. Chat box is not working. Yes, please type. You have to communicate to me, right? We have to interact. Otherwise, it will be like one-way process. I'll just keep on speaking. You'll be just listening, right? No, you have to talk to me. So please type. Few of them have typed. Others also, please keep yourself active. Another 2 hours. You're going to be along with me. You're going to learn. Maybe you must have learned. Let me try to put something extra to the subject what you have learned in the sequence alignment and make it a little bit interesting by giving you some activity also, if possible, time permits. No one wants to speak to me? Okay, it's 6:32. Padma ma'am, I'll start. Shall I start?
Yes, Vanitha.
Okay, ma'am. Thank you, ma'am. So good evening to all. Today, myself is Dr. S. Vanitha. I'm a faculty assistant professor, Department of Biochemistry and Nutrition. So today the topic of interest or the course content which I will be dealing with is multiple sequence alignment by using one tool. Today we will see, which is ClustalW. So many of them have understood or heard about multiple sequence alignment. So we will go into the process of trying to understand it basically by giving you a theoretical idea about what it is, and then we will go to the practical session where I will show you how we can perform it. As a continuation of this one, we will show you another one open resource which is called as Galaxy, where at the end of the session I'll show you. So where you can run the same multiple sequence alignment using ClustalW in Galaxy also. So that's how the work plan will be for today. Hope I am audible to you. So I'm just sharing the screen. Hope it is visible. And so day 2, it is multiple sequence alignment. Hope you are able to see it.
Yes,
If you
ma'am.
say yes, I'll continue. Fine. Okay. So coming to multiple sequence alignment, we are using a tool for that one, which is called as ClustalW. There are other best tools also which are available to perform the multiple sequence alignment. But today we'll work with ClustalW. It is very friendly to handle it, and also the output what you get, it is easy for us to interpret the result what you're going to get from that one. So it is a general purpose multiple sequence alignment program. It can be done for DNA, it can be done for RNA, it can be done even for the protein molecule. Then the sensitivity of this method by which we are going to use a method called as progressive method for this multiple sequence alignment, which has greatly improved the alignment of divergent protein as well as the nucleotide sequences. Okay. The main objective of ClustalW is to align 3 or more sequences. Generally, when the title says multiple sequence alignment, so you require more than 2 sequences. So to start with, we'll be taking with 3, 4 or 5. And now the latest version of ClustalW, the number of sequences what you can do can be up to 1000 sequences also. So what is the objective of this one? Why do you have to do this multiple sequence alignment? Is to find whether there is any structural or functional relationship between the sequences. So if you take a nucleotide sequence, or if you're going to take a protein sequence, you want to compare the sequence and see whether there is any relationship between them. So ultimately, when you perform this multiple sequence alignment, you can get to understand some of the properties or some of the unique features that you can find in these sequences. One is called as the conserved regions. So in biology, during evolutionary time, there may be some regions which are called as which can be a group of bases, or it can be sequence of nucleotides which will be preserved as such for many generations in the form of DNA. And those sequences or the regions which are being preserved for many generations is what you call it as the conserved regions. So when you're performing your multiple sequence alignment, you can predict which regions on the sequence what you have taken are having the conserved regions within them. Other than that, also you can try to understand presence of the consensus sequences also. So if you take a nucleotide or an amino acid sequence, when you're taking among the nucleotide or amino acid sequences within that base pairs, you're going to see that there are certain places or regions or position of an nucleotide or an amino acid which will be frequently particular region, it will be present in many of your sequences what you have taken. So that is what you call it as the consensus sequence. Okay. So continuing to this one, so under ClustalW, there is an tool which we are going to use, which is called as the dynamic programming tool. This is the earlier tool which was being used in order to perform the pairwise sequence alignment. So today the topic is multiple sequence alignment, but initially people were doing the pairwise sequence alignment, where they approach to use the dynamic programming tool over there. So it is like an algorithm which will be running behind, where it will try to take the big set of data and break it into small set of data and try to compare and check out to give you the best solution. So that's how the dynamic programming tool will be running behind. And since there were few limitations which we could not use the dynamic programming, researchers have switched on from the dynamic programming tool into the progressive method in order to align 3 or more sequences. So currently for multiple sequence alignment, the tool which is running behind is a strategy or a tool which we call it as the progressive strategy or progressive method is being done. So this was being suggested by Feng and Doolittle in the year 1987. So what exactly is happening during this progressive? I request the participants to mute yourself.
Please mute
please.
yourself.
So what exactly happens during this one is that first there will be comparison made between pair of sequences. So pair means what? Two sequences. So they either two nucleotide sequence or two amino acid sequences will be taken and it will be compared between both of them. So this is standard dynamic algorithm because it's a pairwise sequence alignment. Okay. So during this time, what we can calculate is the distance between the sequence pairs. And for calculating the distance between the sequence pairs, they have used an equation that is n. N indicates to you the total number of the sequences multiplied with the total number of sequence minus 1 and divided by 2. So when you do this one, you're getting the distance between the sequence pairs, and this becomes the criteria for calculating the distance matrix, which is being put up into the clustering algorithm. So hope you know what is this clustering algorithm. Clustering algorithm means two sequences will be now joined together. You get the clusters, and from that clusters you will be able to check the similarity between the sequences. So initially you start with pair of sequences, then pair two sequence, then with the third sequence, then with the fourth sequence, like that step by step there will be alignment of the sequence. So when two sequences are being checked and compared to together, then the distance between the sequence pairs will be calculated. So this is all mathematical programs which will be running behind to calculate them. And with this clustering, 2 to 3 to 4 to 5 to 6, like that, how many of sequences it has been clustered now, one after the other, right? So finally that will result in the guide tree which you will be getting out. So this is the initial stage, what happens during the program of ClustalW or multiple sequence alignment is being done with the method what I told you called as the progressive method. So how do you start for progressive method to do multiple sequence alignment? Initially you start with two pair of sequences, then the next pair, then the next one, the next one, like that step by step the sequence will be aligned. Okay. So when you put all these things, what you're getting now, you'll be able to construct one guide tree. What is the word guide tree means? It means that it is trying to give you the relationship between the sequences. So you take two sequences, if you're able to get a guide tree that is telling to you between those two sequences, how much of similarity is there, how much of the distance is there similar or there dissimilarity or any basis you are able to get, or there are gaps in between them, all those small informations with the guide tree you will be able to get. Now what next? You've got the guide tree, right? So once you get the guide tree, then the original process starts. So then it will be aligning the first node with the second node. That is nothing but the sequences what I told you. Then it will fix the alignment. So whenever alignment is taking, if you start from 1 to 100, in many of the times the sequence alignment will not start for all the sequence starting from 1 to from the first. It may start from first, for some it may start somewhere else later also. So it tries to align the two sequences where there are best similarity between them. So it will start to fix the alignment. So first one with the second one, then the third sequence, like that step by step it will start to do the sequencing, alignment of the sequences first. So when all the sequence one after the other is being aligned, you're getting a cluster now, or a group, you're getting group of your aligned sequences. Okay. So after that, when the final alignment is over, then it will calculate and give you the highest alignment score. And based on the symbols, what you get during the alignment, you will be able to understand about the sequence. What are the sequences you have taken, whether there is similarity between them, anything there is a gap between them. There is an interpretation of all your sequences. The purpose is to check whether there is a relationship between the sequences what you have taken. Okay. So you take pairwise sequences. you compare them, then the third, fourth, fifth, like that, step by step, the progressive method is performing to align one sequence with the another sequence. And finally, when all the alignment is being done, you will be getting your guide tree. What is the meaning of the word guide tree? Guide tree is telling to you the relationship between your sequences, what you have taken for your study. Okay, so once you do this one, finally you will be getting your output. We will see that one later. So it is Clustal Omega is using progressive method or algorithm by adding the sequence one by one until all the sequences are being completely aligned. So this is the steps with which you will be performing the Clustal program. So first you will calculate all the possible pairwise. You are not doing it. The tool will be doing it, but I'm trying to give you what exactly is running behind. Okay. So it is able to calculate all the possible pairwise alignments, record the score for each of the pair, and then first it will construct the guided tree based on the pairwise distances. For this one, the algorithm which will be running behind is called as the neighbor joining method. Then find the two most closely related sequences. That's what the job is running behind. Then align the sequences by progressive method. Then calculate the consensus. Then replace two sequences with the consensus. Then find the next most closely related sequence. Then iterate until all the sequences have been aligned. Then expand the consensus sequences with the gapped original sequence and report the multiple sequence alignment. So this is how the original program will be running behind in the Clustal Omega. Okay. Now this is giving you a summary. What today I'm going to use the tool for you to teach the multiple sequence alignment is called as the Clustal Omega. But other than that, also we are having various other best multi-sequence alignment tools available. Okay. So first one, as the name goes on, it's ClustalW or Clustal Omega. Here it will use very less amount of memory only. And the caution, what you have to take when you're performing Clustal is it should be scalable than the modern programs, because you have a list of other programs are also available for the multiple sequence alignment. Then die align, which is attempting to distinguish between alignable and non-alignable regions. It's less accurate in giving the output when compared to that of ClustalW. MAFFT, MAFT and muscle, they are faster and the output is having more accurate result when compared to the Clustal Omega or other tools. But it can be used only when your data set is large. Just if you want to do with 3 or 4 or 5 sequences, then there is no purpose of using this particular tool. So you can go with less number of sequence alignment with ClustalW. But in case if your data set is very large, it has to run very fastly, right? So for that one, you have to go with MAFT or muscle, where it will be doing even the sequence alignment for 1,000 set of sequences. Then you have pro cons, which will give highest accuracy score, but the caution is computation time. So when the sequences are less, the output also will be obtained very quickly. It takes very less time only. But in case if your sequences are more, then it will take more amount of time. And the another caution or the factor which can be limiting factor for this particular program is the memory usage, which can be another concern for us. Then you have pro DA. Here it is no global alignment and the sequence will be repeated, shuffled. So a lot of different strategies will be running when the program is being run. And here the caution is high computational cost and less accurate result than ClustalW can be one of the caution when you are using this tool. This many of you must have heard about it, which is called a tea coffee, which is highly accurate, or accuracy is very good with this strategy. But our main concern is for the computation time. That's the output, how much time it takes and give you the result for interpretation, and the memory usage are the little concern for us. So this slide is explaining to you about the multiple sequence alignment. The other programs other than ClustalW, I have tried to give you the advantages and the cautions what you have to take in this one. Okay, so hope I am clear till here. Yes?
Yes, ma'am.
You can just type Y in the chat box. I've kept the chat box open. Yes, yeah, just put Y. Y is enough. Capital Y is enough. If you're putting capital Y, it means you are listening to me. If you put small y, you're not listening to me. Yes, I find how many participants are there for today? Might be 30, but response is only from few. Okay. Till now, what I have told you, do you have any points to be repeated or to be dealt, or shall we go to the next part of the slide? Shall we go to the next part of the slide?
Yes, ma'am.
Yes. Do everyone accept? So no doubts till now? You are not having any doubts? Steps in ClustalW, can you repeat, ma'am? I will show you itself. That is like a program which has been running over there, but behind program. But originally, when you take the tool and when you're running it, I will show it to you. Srinidhi, I will show it to you. Yes, okay. So now I'm going to show you the continuation of this one. I hope you are able to see the slide. Okay. So now we are going to see the uh, how to use the tool and get the output or the result, and how to interpret. Generally, what we observe is that when you say open the Google, take the site, and if you give the link, they'll just type it or do it, and then execution is very, very good, excellent, because students or the participants or whoever it is, we perform the I mean, we are very quick enough in adapting to the technology and we'll start. But the problem comes what to do with the output, right? So let us see that till the output, what you can do when you want to do your multiple sequence alignment. Okay. So just in your Google, it's a very good, faster search engine. So there you can type the word as Clustal and put the word also as omega if possible. Otherwise, just typing Clustal is also okay. Okay. So if you, I'm step by step, I'm giving you in my slide also how the site, how it will open up and what you're going to do. Keep your eyes wide open, keep your ears very crystal clear, because at the end, I'm going to ask you to do something, you're going to do, and you're going to show me the result. There is no any escapism because we have already told that you'll be having an assignment by each of the faculty. So if I dealt with 5 different topics, so I'll be asking you to do some assignment or a small work on Clustal Omega using this tool. So you are supposed to do it. So please keep your eyes open, ears also. You have to listen to me very clearly. Okay. So this is Clustal Google. You can work along with me. You can now open your tool in your laptop or your system and you can do. It's a open free software only. So there is no any downloading for this one. So directly you can use it. Okay. So now in the Google, once you put your search, ask your search engine, it will give you the output like this. So when you put Clustal and Omega, you can see there are so many details coming out of it, the tools which are available. But I request everyone to go with the first one, what I have shown over here. That is, you can see the first one. I'll just take this. Yeah. So now you can see here, this is EMBL, EBI. So yesterday you studied about EMBL, right? So this is the database center where they are providing this tool, that is a Clustal Omega. Now you go with this one only, see for the site properly and use only this particular one. Okay. So once you click this one, you will get the home page of the Clustal clustal omega, which will be exactly looking like this only. So what are the points to be kept in mind when you are opening the clustal omega tool? Now you can see there is a first query asked, which is called as input sequence. But before going to that, you can also read the data that is given under clustal omega. It's a new multiple sequence alignment program.
Vanitha, screen is not visible, it seems. Screen is not visible. Somebody put a message.
Is it the same problem with everyone? Now?
No, ma'am, it is
It is visible, ma'am.
Okay.
No, somebody has put a message saying it is not visible. Now it's visible.
Now it's visible?
Yes,
Now
ma'am,
is
it
it
is visible.
Okay, ma'am. Thank you. Good. Okay, so that uses the seeded guide trees. Just now we have seen all the points about the guided trees and HMM profile techniques to generate the alignments between 3 or more sequences. See the word, it is always between 3 or more sequences. For the alignment of 2 sequences, please you can use a pairwise sequence alignment tool, not this one. And here you can see this particular tool can be used to align more up to 4,000 sequence, or the maximum file size can be 4 bases. Okay. Now, so below this, you can see that you have the first 3 data will be provided over here. So accordingly, you have to give the input and select the parameters if required, and then go with the submit. Okay, it's very, that's why I told clustal omega is very friendly to us. So here you have the first one, which is called as the input sequence. So here it is asking you what sequence type. It can be a protein, it can be DNA, it can be RNA. Okay, so here you have to select. So what is the tool? The tool is clustal omega, but what type of a sequence you want? Is it a protein sequence you want to make a comparison, or you have to, you have to use a DNA or RNA? So if I click protein, then here in this particular place where you have to paste is the sequence. Okay. Otherwise, in case if you have the accession number, yesterday you have seen all the database centers. Every database center will be storing the data which everybody submits by giving one accession number. So that accession number also you can take it and you can paste it in this box or the window, so that you will be able to run. So point number 1, input sequence. The input sequence, it can be a protein, it can be DNA, or it can be RNA. Point number 2, remember in this box you have to paste the sequence, or you can give the accession number. I'll give you the third option also. What you can do is that below you can see over here, which is called as choose the file. So what you can do is that you can go to the database centers. From there, you can download the sequence, nucleotide or a DNA or protein sequence, or your RNA sequence, and save it in a file in your computer, in your tool, that is in your normal laptop or your system. You can save it, and then instead of pasting the sequence, instead of pasting the accession number, you just click choose the file and upload your sequence directly. So three ways I've told you. One is to paste the sequence, 2 to give accession number, 3 download the sequence and you can upload it over here. Now, very important point when you are inputting or giving the sequence over here must be in the FASTA format only. This is very, very important. As you know that FASTA format, how it will be present, you'll have a greater than symbol followed by the accession number and details about the organism or what type of a sequence it is, like a linear DNA or it is an RNA or it's a protein. All those data will be given in the first line followed by the sequence. Okay, so remember in the input 3 plus 1, 4 points I told you. First one is type of the sequence, protein or DNA, RNA. Then you have to paste the sequence or give accession number, or you can download the FASTA format and you can upload over here. And then the most important last point is that the data what you're going to give as input must be in the FASTA format. Okay. Now, the next thing what you are going to see over here is going to be the parameters. Okay. So the output parameter, how you want also it will be asking. Maybe if you just click this one scroll down button, you'll be getting lot of parameters. So we will go with ClustalW parameter only for today and check out. If you want to experiment, you can use even the other parameters also and check ClustalW when you use what is the output. When you use ClustalW with character count, what will be the output. So you will know the difference how the output is coming when you're running this program. Okay. Then finally you have the submit button. In case if you want to give a title for your work what you have done for today, you can do it. Otherwise directly below you have the button which you call it as the submit button. So just you can give submit and you can just wait for some time and you'll be getting the output. Okay. So this is how the Clustal Omega, the tool is and what we are going to do in that one. Okay. Now coming to the next aspect of your multiple sequence alignment is to retrieve your sequences from the databases. Yesterday you have said about NCBI, you have seen NCBI, EMBL, you have seen DDBJ. So any of these tools you can use in order to get your sequence. Okay. So today I'm showing you with NCBI, then we will work on with others also. So just I have make it very simple, taking here from the all database, just scroll down button, select the or database what you want to. Let me assume I've taken the nucleotide sequence for which protein, cry protein. Okay. So many of you know that it is Bacillus thuringiensis gene which is present in that organism. So now after giving this, putting the search button, I'll be getting the list of all the information. Okay. Now here I just got here the data. So I'm just taking here. But what I told you, I want the sequence in what format? FASTA format, right? So you can see in that when you're working, if you're working anybody of you can just see a one tool called as a summary. You have the scroll down button, just click that one. You'll be getting the, how to take the data and which format you want the data. It will be coming out. So you can select the FASTA text format for this particular tool running program. Okay. Now again a small window opens up where it will give you the FASTA format of all the list of cry proteins that was available in the NCBI site. Okay. So just now I told you have the greater than symbol, the accession number, then this is Pisum sativum, that is a plant, that is a cry. And here it is showing you the mRNA sequence and complete CDS. CDS is the coding sequences. Okay. So now what did I do? This is the first data, first sequence I got. Just you copy it and you can paste it. Did I tell you what you're supposed to do in the input side? Right. You can just paste it over here. Then again, go back to your FASTA text, select the next one, paste it. Okay. Now when you're pasting it, be very careful. You have to make sure that this first sequence is ending over here. Then you put the cursor here, put enter, see that the next line immediately you will be having here to paste your next sequence. You cannot put the paste here itself, it will not read. So how do you paste is also important. Always when I'm running the program, I'll show it to you. Okay. Then this is the next one which we have copied it and pasted here. 3 sequences I've shown to you. And after that, you can just put the word as submit over here. Okay. So once you do that one, it will be running, the program will be running behind, and since we have taken 3 or only 4 sequences within short span of time, you will be able to get the result. So the tool will be showing you like this. So just click view result and you will be getting the output in this format. So you can see I have just highlighted here. This is the tool output. That's what it's showing to you. Then you have the next one which is called as the alignment. This is what we require, right? So what do we require? You have taken 3 set of sequences and then you have aligned them one after the other. So So when you click this alignment, then you can see here, you can see the accession number and you can see all the bases that are being aligned over here. Okay. The next tool is called as what? Guide tree. What is a guide tree? I told you it is trying to show you the relationship between the sequences. Then there is one more add-on result what you can get with this tool, that is a phylogenetic tree, which will try to tell you the evolutionary relationship between the sequences what you have taken. Okay. Then result reviewer is a result file, submission details will be there. If you want, you can download the whole output and you can save it also for any of your purpose. Okay. So now what is a thing is that now you have to interpret your result, right? So you got alignment. So you can see the alignment, how it has done. So you can see on to my left hand side to this end, you're seeing here, right? How many sequences I gave? Only 3 sequences which are placed one below the other. And you can see the bases are being starting from here till here. Okay. Now, what do you find here? You find some dashes. What does that indicate? It is called as the gaps. Okay, so then you have the second sequence, then you have the third sequence. Now, once you get the alignment, you have to look for 3 different data to get over here. One is to see below the 3 aligned sequence, what do you find over here? Here I can find some asterisk. Here also you can find the asterisk. Here also you can find the asterisk and asterisk. Now what does this asterisk indicates to you? Just observe the column straight to this asterisk. So here you have the asterisk. Here I have A. Here I have A. Here also I have A. This is first one. Second asterisk you look G, G and G. Next asterisk you see A and A. Next asterisk T, T and T. So now what do we understand from the output when you are aligning the sequences? First one is look for your conserved regions. So now wherever you are able to find the asterisk or a star mark, it indicates that in that particular position, those bases are said to be highly conserved regions. Okay. So like this will be a first output. Since this is nucleotide sequence, I am getting the output only with the asterisks. With amino acids, I will show you what will be further the output, what you will be able to get shortly. Okay. Then you can see here, this is what you can see here, same nucleotide sequence. I've colored it also. How to color it? We will use the tool, that time you can see it. So now you can see here, they're not only asterisk, you got something else also over here. Okay. So one is this colon, you can see here. So now just have a look towards this colon. Here you see column, you have to see straight above the colon. You can see this position is A, here it is A, here it is T. Okay, so 2 are adenine, 1 is thymine. Now here again you see, here again 2 are adenine, adenine, 1 is thymine. Then again you have here colon, it is adenine 2 and thymine. Okay, then you can see here it is A, 1A and 2T. Okay. So this is what is you're getting here, which is indicating to show you that adenine and thymine, they are purine pyrimidine bases which will base pair with each other. Okay. Then coming to another region where you have this single dot, you can see. Now you just have a look at this particular column. You have guanine, guanine and adenine. So all the 3 are said to be as what bases? Purine bases. Again, we'll see, do you have here, you have one more dot. Just to see across that column, you have guanine, guanine and adenine. Here you see you have cytosine 2 and then you're having one adenine. Completely they are not complementary to each other. Then you have here 2 more dots. You have guanine, adenine and guanine. No possibility of pairing. Then you have here C, that is here 2C and 1A, that is one is purine, the other 2 are the pyrimidine bases, not TT but CC. Pairing is not possible. Okay. So like that, you'll be able to get the result which you can compare and tell. Okay. So here when you're taking about asterisk indicates about you the similarity. So you can see that all are the, in that particular position, all the bases are the same. Okay. So next one is guide tree. So if you click that one, you'll be getting the output. So 3 different sequences. And after this .1.1, this till here, it is telling you the accession number. Now this is the distance score, what it has got when it has compared one sequence with the other sequence. Okay. So this is guide tree, what you will get. That is only trying to give you the relationship between the sequences. Then this is only for bases. I have tried it. Now even for proteins also I tried. This is called as a prion protein. You must have heard about it. Same way you have to do it. So now you can see the output for the protein. Here also you can see I've taken 3 different proteins and the alignment is done over here. And for your convenience sake, I have put the amino acid chart also with 3-letter word and single letter word. Here it is always only a single letter word in the output you can see. Now, as I told you earlier, you can see so many number of stars or asterisks, and you can cross check all your columns. You can just check out each and every column. Wherever you have the asterisk, it's all the same amino acids. WWW is tryptophan, GGG is glycine. Okay, now you come to the colon, what you have here. Now it is 2 of the sequence in this position are showing L, 1 is showing you F. Now what type of an amino acid it is? Because we immediately, if I ask single letter word, it's difficult to interpret, but you can look into the chart and see what type of an amino acid it is. If it is single letter word L, it is indicating leucine, and if it is F, what type of an amino acid it is going to be phenylalanine. Okay, and why it is given a colon? All are not phenylalanine. All in this position are not leucine, but 2 sequences are showing leucine and 1 is showing you the phenylalanine. They are being grouped here as and represented as colon, indicating that they are the amino acids which are having the same size or same hydropathy properties. That is, as such, when I compare phenylalanine with leucine, you know that they all are hydrophobic amino acids. Okay, so when you have such type of comparison and you get the result with the properties matching with them, then you will be getting your colon. Okay, and here also you can see the colon. II stands for isoleucine, L stands for leucine. All are branched chain hydrophobic amino acids. Okay. So the same thing, you can select the color and you can get the colors for your amino acids also. Very systematically it will color, and by just seeing the color also you can tell whether they are conserved sequences or bases or the amino acid present in that particular region. You can compare it and get the output. Okay. The next one, for the same protein, we have checked even the guide tree also, and we have got the output. And as I told, interpretation of the result. So once you take the output, you will be getting 3 different outputs. One is asterisk or a star, which indicates it's an entirely conserved column. Column means you're looking straight, right? So that one. Then if you get a colon, it indicates that it is a column where all the residues have approximately the same size and hydropathy properties. And if you get a single dot, okay, it indicates that in that particular column where the size of the hydropathy has been preserved in the course of evolution. Okay, so that is what is the output you will generally get after aligning 3 or more sequences under ClustalW program. Then criteria for building is to check the structural similarity, evolutionary similarity, to get functional similarity or the sequence similarity. And the main applications of multiple sequence alignment is to perform phylogenetic analysis, pattern identification, domain identification, DNA regulatory elements, as well as the structure prediction. So these are the main applications where you can use your multiple sequence alignment. What we are doing is very less number of sequences to initially try to understand what the output is, how it is, how to read it and get it. But when you're performing some research, maybe during that time when you're working with many number of sequences, so you should have an objective or an aim. So any of this you can keep your aim in order to get the outcome or the output after your ClustalW. Okay. So that is about what I thought I would like to explain to you. So now if you have any doubts or queries, you can ask me. I just now told the difference between colon and dot, right? So colon is trying to give you the size and hydropathy in that particular column will be the same. Okay. For example, if you take amino acids, leucine, isoleucine. Okay. As such, when you check, it is not same. It's not isoleucine, but it is isoleucine, and leucine. They all are coming under what category of amino acids? They are the hydrophobic amino acids. The dot indicates to you that for example, I take an amino acid. Let me assume that that particular amino acid, its molecular weight is 20, for example. Okay. When I take that sequence, the amino acid may be different, but when I take the another sequence and compare it in the same position, and another amino acid is it, but its molecular weight or its size is only 20. But they both are different, completely different. But the size is the same. They are again hydrophobic also, but it is completely different. No matching between them. So it's that indicates to that it is highly preserved region to occupy that particular region with that particular size, a nucleotide or a base or an amino acid in that region. So that will be represented with a dot. Okay. So if you have understood, just put capital Y. Chat box. Okay. I have nearly, if I exclude myself and the other members in my team, I think I should get 53 responses. I am waiting. So only few are active. What the others are doing? Huh? Okay. So now the theoretical part, how to run, I have just shown to you. But if you are okay with it, I'll go ahead with sharing the screen to show you to run the program also. The tool also I will show you now. Can we go ahead? Put A in the chat. Put A, capital A. Listen. Keep your, yes, A. Yeah. So I'm going to go ahead, show you how to use the tool. Okay. Thank you all. Thank you for your responses. So we will just go ahead with this one. I'm just sharing the screen again to you. Okay. So this is the, okay, to make it. So first thing what you're supposed to do is what? You have to take the search engine and type over there what? Hope you're able to see the, you're able to see, right? I've shared the screen. Yeah, thank you. So just type and select ClustalW. As I told you, you'll be having the programs in different formats given over here, but I want you all to try only with the first one, that is EMBL EBI site in order to run the ClustalW. Okay, so I've just given here. So now, as I told, you can see what and all I told, the input region sequences. You can see the output format it is asking. Just open up, it asks ClustalW with the character counts W, then FASTA, MSF, or all the other things. It's showing you the parameters. Okay. And then you finally have the submit button. Okay. So now I have opened the tool, but now I have to paste the sequences. Okay. So how do I do the pasting of sequences? How shall I do that one? I
By
read
retrieving
one.
sequences from NCBI.
Okay, fine. So you want me to use NCBI? Fine. I'll use now NCBI. How to hide this one? Fine. Okay. Okay. So I'll just go to Google and let me show you another. Yeah.
Ma'am, can you use another one instead of NCBA?
Yeah, that's what I wanted to do. I will go with today with your tool that is DDBJ. What is DDBJ?
DNA
DNA
data
data
bank
bank of
DNA
Japan.
Yeah,
data
fine,
bank of
fine. Now, how to retrieve the sequence from DDBJ? Anybody can tell me the steps? Yes?
Search for a
Okay.
target of interest.
Yeah, search, fine. Then you can go here, ARSA is given. You can just click over there. Okay. Then you can type your, I have typed pyruvate kinase earlier when I checked. So you can search. Okay, I got some output. So you can see the result what I have given here. So you have here accession number is there, and you have what type of a protein. Now since it is pyruvate kinase, it's a gene. You can see the sequence length. Its molecular type available over here is the DNA. Okay, and it's a synthetic construct. So all the details of that particular sequence, what has been deposited in DDBJ is shown over here. So it's a flat file format. So I will require what format now? FASTA, right? I cannot use all the files. You have to go only with the FASTA format. Maybe what we will do is that we will not take, if I just go ahead down, you will have list of pyruvate kinase related information. So I can't run such a huge sequence. So what we will do is we'll take only few. Maybe I'll select 1, 2, and 3. Just click it over here. And it's asking if you want to view the selected. No, I don't want. You want to download it. Maybe I'll download only the selected 3 entries. Now for this downloading, you can use the FASTA format only. Okay, so let me click and see download the selected data. Let us see what it is showing me. As I told, it is downloading the sequence. Okay, but when you are downloading it, you have to make sure that where you save the data, so that when you go to the original tool uploading the data to retrieve the sequence, you should know about it. So what you can do is that I am at present, for our convenience, I am selecting the downloads to save this one. So I'll just save it. Okay, so now my data is saved not in the form of a Word document, no Excel sheet, but it is a program or a document. It's just in the FASTA format. Okay, now I have got the sequence for pyruvate kinase from DDBJ. Now I'll go to Clustal Omega. So since it says to me as this is DNA, the sequence is DNA. Okay, so this is RNA. I don't know for third one, but we will just try it out. So I'll just take DNA. Now what I'm supposed to do, I did not copy the sequence. I did not take the accession number, but I have downloaded the data. So just go to choose the file, and directly it is taking me to the downloads, and it is saying, yes, you have the sequence, what you have downloaded. Select it and open it. The moment I have done it, you can see it has taken up. So let us try to go with ClustalW. Initially, I'm selecting the parameter and we will do submit. Submitted, as I told, it is working out, but it is telling invalid parameters. It's saying that sequence, there is error in reading the input sequence. Please check your input. Okay. So what we will do is that maybe it is a huge nucleotides. It's only 3,602. Sizes are too big. Okay, so we will go with the other way around of taking the sequence. Now what you do is that because it's a huge data, maybe it is not able to get through it because the format in which it is, I have downloaded FASTA only, but still I will show you the other way also to get the sequence. To go to a get entry, and it is asking me to put some number, that is accession number. Now here also when you're putting the accession number, You have DDBJ, EMBL or GenBank. So this particular is taken from the DDBJ. So just take only the DDBJ accession number. If you're taking the accession number from your NCBI or in GenBank, then you have to select that one and you have to put it. Okay. Now here also it's a flat file. You have total nucleotide sequence. You have the FASTA also. So this is amino acid sequence FASTA format. This is nucleotide sequence FASTA formats. You can, let me see if I take amino acid sequence, what will I get? So I got the amino acid sequence for pyruvate kinase. So what I can do is that one option is to copy the sequence. Let me show you this one, then we will sort out the other thing. So So I have pasted the first sequence. As I told you, here the cursor is blinking. So what you do is you put enter, okay? And you can see that I did not take nucleotide sequence. At present, I've taken only the amino acid sequence, okay? So I will try with some other one also. That one was, so I will take another one. So instead of this one, I can paste the another one. I hope it is different. I'm selecting the same one, coding sequence, amino acid, FASTA format. So I'm getting, did I do the same thing? This is what? Saccharomyces. Yeah, it's
are same, right, ma'am?
Yeah, I'm changing it now. I'll go with the next one, soya bean. Okay. This is soya bean. Let me see. Okay, this is glycine max. You see, you have to take it from the greater than symbol. Okay, and then you can paste it as I told. After this, you have to paste it. Okay, linking, press enter. So I took for soya bean and now I'll take for trichoderma pyruvate kinase and then paste it in the DDBJ. Paste it, search, same amino acid sequence, FASTA format only I'm taking it. So at present I've taken 3 set of protein. So I put a cursor. Okay. So instead of choosing at present, I have just copy pasted the sequence to initially start with. Then we will sort it out. Okay. So this is ClustalW. I have selected submit. Hope I get a result out of it. So your job is running. So maybe after 2 or 3 minutes, I may get the result. So I got the result, view the result. So this is the result. If anybody simultaneously running, mostly must also be able to get it. So I have taken glycine max, Saccharomyces and trichoderma. I gave Saccharomyces first, I gave glycine second, I gave trichoderma third. But you see how the sequence has been aligned. First it has taken glycine max, soya bean, then Saccharomyces followed by trichoderma. Okay, so that is how the algorithm is running behind to take the first sequence, which is having more sequence similarity with the other one and comparison ordering of the sequences. Okay. Now you can see you have the accession numbers and this is the organism from where we had taken. You got the output. If you want, you can download it. Otherwise, I'm saying I want to seek the colors. So just below, if you go, you'll have the alignment with colors. So you can color all the sequences. And now let us see the output or the result. I don't have amino acid list with me, but if you are able to guess and tell me, it will be fine. So now I have here, let us see first the asterisk. So just to see the column ahead, what is the amino acid you're able to see in all the three, in the same position, all the three amino acids are which one? L. L stands for what amino acid?
Leucine.
Leucine. What type of an amino acid it is? It's a hydrophobic amino acid. And asterisk, if you get, what does that indicate to you? That it is the conserved amino acid in that particular position when we compared these three sequences. Okay. Then you come with the colon next. Okay. What do you find here? Two of the sequences are showing R, that is arginine. One is showing K. What is K?
Lysine.
Lysine, exactly. So what is the, what is arginine and lysine under amino acids? We classify them into different types based on, it's a basic amino acid. That means on the side chain, they'll be having the arginine contains guanidino group and the lysine contains the epsilon amino group. Okay, so they are basic amino acids. So that's why you have the colon indicating that in that particular region, you can have the amino acids where its size as well as its hydropathic properties are the same. Okay. Now you see this colon. What do I find here over here? Is there any relationship between these amino acids? Only one relationship is there if I'm not wrong. What does V stand for? It stands for valine.
Valine.
Yeah, L stands for leucine, F stands for phenylalanine. Yeah. So when I take all these three as per hydropathic thing, you can see that they all are said to be as hydrophobic amino acid. Okay. So it has been confirmed that in this particular position, you can have that amino acid which will be having only that property called as the what property? Hydrophobic property. Yeah, but the amino acids will not be the same. They will be different. So like this, if you check as you go ahead down, many places you can see highly conserved regions. You can see there are many different regions. That's why I colored it so that you can see the column. There are a lot amount of sequences which are the same only. Okay. So this is the first one, alignment, job done. Then you can go to the guide tree. So you can see the guide tree. First, you can see here a bracket and then the accession number, number of amino acids, what you have taken, glycine max, and this is the distance, 0.31746. That is the mathematical algorithm. What I told is running behind. Then you have this DDBJ, then you have the, this is DDBJ accession number, then this is accession number, then you have the number of amino acids and Saccharomyces, and this is 0.1953, and you can have the last one also. So if you just go below, you can see the guide tree. So now you can see the distance present between them, and you can see that when I take Saccharomyces and Trichoderma, according to the algorithm output or the result, you can see that the distance is very 0. I can say it's almost the same. That's why they have one single node and you have the two mean positions which they are arising from one single node. It has been arising. That means the sequence are very much close to each other when you compare them. But the other one, the distance is very far away from them. As you see, the distance is 0.317. So that's why from the node it has been kept away when you are comparing the three sequences. Okay. So what is the use of guided tree? What is it trying to give you is the relationship between the sequences, right? Now come to phylogenetic tree, same data. So this is trying to give you the evolutionary relationship. Now you can see the distance over here, glycine 0.39401. So this is the distance. These are all your accession numbers. This is Saccharomyces 0.19821 and Trichoderma is 0.174. So you can see that in the guided tree, when you're keeping two sequences, they were very good pairing is there between them. But when you take the phylogenetic relationship, they're completely different. Now you don't look into the distance, look into the species what you have taken. Glycine max, soya bean, you have taken a plant. Saccharomyces is a yeast, Trichoderma is an actinomycetes. So all three are different type of organisms. They are not the same. Okay, so obviously you'll be having some difference in the distance. But when I compare all the three, you can see that Saccharomyces and Trichoderma is little bit close to each other when compared to that of the soya bean. Okay, so with this one, you will be able to get the phylogenetic tree. So as I told, you can view the result, then result files and submission details, you will be able to get it. And if you want to download, you can just go to the alignment and you can download the data. Yeah, this is again alignment. Each amino acids are given different colors, so you can magnify them or you can zoom out or zoom in and check out all the amino acids in the positions. And you can see RR, all are R indicates arginine, G indicates glycine, and H indicates to you histidine, S is serine. Okay, so like that, M is methionine. E might be aspartic acid, if I'm not wrong. I have to check it out. So like that, T is threonine. So you can expand here and check out the positions here. You can see here. So the ranges you can expand it or you can reduce it, zoom out, zoom in, and check out the positions separately. Earlier, you just, yeah.
Ma'am, if we take two sequence from the glucokinase and two sequence from the hexokinase, if you compare
Again, you
both
tell
of
me.
them, what will we get, ma'am? If
Tell
we
me
take the four
again.
sequences, if we take the four sequences, ma'am, from
Okay.
two are from glucokinase and two are
Okay.
from hexokinase.
Yes, yes.
What will be the result in the
We can check it out.
and polygenetic tree?
We can check it out, right? Can we check it out? You can check it out, right?
Okay, ma'am.
Yeah,
Okay,
check
ma'am.
it out and give me the output. That's a good question. That's a good work. What we can do now. So why you have chosen? Maybe I may think that hexokinase, glucokinase, they both are glycolytic enzymes. Okay, hexokinase can act on all the 6-carbon sugars. Glucokinase is highly substrate specific. It acts only on the glucose molecule. Based on the Km value, you can find that glucokinase is very specific towards the glucose molecule. Okay, so maybe if you take it out and check out the protein sequence of both the kinases, we will be able to see whether there is any sequence similarity and predict what is the outcome of your omega, that is your multiple sequence alignment. We can check it out. Can I know your good name, please? Whoever is trying to work it out? Hexokinase and glucokinase? Mahesh. So I'm waiting for you, Mahesh, to show you me the result. Okay. So hope you have understood. Yeah. Hope you have understood how to use the tool, right? Right. Yes. So, so now what I'll do is, okay, you're responding. That's nice. Okay, good. Yes, absolutely. I hope you all are working. Hope you're all working. Yes.
Yes.
Yeah. Yes, good. Yes, good. Yeah. Now, yeah, good. Nice. That's good response. I'm very happy. Yeah, happy. I'm very happy. Yes, thank you so much. So NCBI, I'll just give you a brief idea how to customize and retrieve and take a sequence also. So maybe I'll just give you a tool over here. I've opened NCBI. Maybe I'm not sharing the screen to you. Okay, I'll just show you. Okay, so I have shared the screen. Hope you're able to see the screen. So NCBI I have taken here, okay, because it's very commonly used from all databases. At present, I have taken protein. Then to make it easy, my job, I'll just go with the cry protein. Okay. You can see now I have selected a protein, a sequence what I want for the cry protein. Okay. Now I have the output. Now you keenly see the output. Each one page in the output contain how many items? It will show only 20. So if you just count, you will get cry protein, 20 data will be there. Next from here, you have to go to the next to get how many set of proteins. Here it is showing 1389. So you have to scroll down to 1389 pages to get how many cryprotein data that is deposited over here. 27,776. It's a huge data set. Okay. So what you can do is that I know that cryprotein is from Bacillus thuringiensis, right? So you can customize to take your data. What do I mean by the word customize? You just see onto this NCBI to my left side, you have your species one set. You can just click there over there, customize, and you can select whether you want only in plants or in the bacteria, you can select it. Okay. Now you remember, you see the list of all animals 802, plant 133, fungi 170. You see the list of data here, and 27,000 some number of output we have got, right? So what you can do is that at present to customize, I've taken only the bacteria and I've asked it to show. Okay. After that, you can go to your sequence length, or you can go to your sources here, and you can select PDB or F sequence or UniProt or DDBJ, EMBL, GenBank, PIR from anywhere. Let me put, for today, I'll put it as PDB, Protein Data Bank, and then try to get the sequence. Okay. And genetic compartments also are there. Sequence length is there, molecular weight, release data, revision data, all are there. Okay. So here you can select like this your species, and you can try to get only from that one. It's not changing yet. Okay. So now I have selected like this. You can, this is one option what I'm showing to you. You can customize it. Then the next one, I told you that you want the data in what format? A FASTA format, right? So you have so many set of data in different formats available. So here I told go with FASTA text format. Okay. So you can just click this one and you can take your data. So I didn't do it. I just gave class FASTA. You can see the window opened up with all the list of that protein in what format? FASTA format. Okay. Now this is one way how you can take it. Otherwise, to make it very simple, let me tell how you can do. Now you just see here cryprotein 808 amino acids are there. Maybe I'll tick this one at present. Then this is Pisum sativum. Okay, I'll select this. Then I'll go with this 1177 amino acid protein. I've selected only 3 cryproteins amino acid sequence, and then what you do to my right hand side, you can see here send to. So click this one. You can send it in the form of collections, and or you can put it in the form of files. So you want the data in what format? Is the file format called as the FASTA. Select the FASTA over here and you can say create the file. So what it will do is that it will for me it is going to downloads, and the sequence, how many I took? Only 3 I took, right? So 3 sequence in the FASTA file, you can save it. I'm saving it in my downloads. Okay, so the sequence has been downloaded and saved in the FASTA format. Okay, so you can go to your Clustal Omega. So now I have taken protein sequence only. So now you can choose your file. So you can see here you have the sequence in the FASTA format. Open it. Now you can see it is, as earlier it was no files, but now it is showing you the sequence FASTA format. Okay, already output is in the parameter ClustalW. So you can just go with submit. So if you submit it, hope it is running. It is in the queue. Maybe all of you are working with all your samples, and this is the output what I have got here. Okay. So instead of copy pasting it, you can also directly download the sequence in the FASTA format, and you can just upload the sequence over there. Okay, so you can download this one and take it, or you can color it, make it a little bit interesting for you to check the sequences, and based on your conserved, or is there any relationship between the amino acids present among the sequences, you will be able to interpret it. Okay. So that's how you can use this particular tool. So this is the guide tree. This is the phylogenetic tree also. The output you are able to get it. Okay. So how many of you are using large data? You can use the same ClustalW, but it may take lot amount of time. So I told you that for your large data set, you can go with Procons. I told you previously in the slide, I explained in the beginning. You can use that one, or tea, coffee. You can use MAFT or Muscle are there, which are very good programs or the tools for large data set, thousands of sequences. You can use that one. See, Clustal Omega is an open source, so there will be always a limitation in inputting the sequence and retrieving the data for it. So for learning, understanding process, we can use the ClustalW. But for research point of view, you have to take some of the other tools and try to experiment with that one. Likita, I think I answered you. Still, if you have any queries, you can message to me. Okay, is anyone trying? Or anyone tried? You got the output? If you are possible for you to share the screen and share it with everyone, fine. I'll be happy to see that that is the outcome of our course, right? You should be able to retrieve, you should be able to analyze, interpret, and share with us, right? Yes? Who is the first person to uh, share and show me? Yes, Likita. Likita, sorry, I thought it is Nikita. Yes, anyone? No one? That means you're not able to, and if anybody wants to, ma'am, I want to learn, please, I'm sharing the screen. Please, you can help me out. Learn to one, if in case if anybody is there, I'm always, it's always welcome, right? It's like a training process. It's an understanding learning process, right? So you can share the screen and step by step, I can help you out. Jay Mahesh, what is your status now? Did you compare, get 4 sequences for your hexokinase, glucokinase? What did
Yes,
our
ma'am, I got it.
ClustalW talk to you? Did it
Yes,
give
ma'am,
any
I got it.
result?
Yes, ma'am, I will share you.
Fine. So one assignment will be on multiple sequence alignment. So I'll give you some sequences either in the form of accession number or a protein name or a nucleotide sequence. You may have to download it and you have to show it. Very nice, wonderful. What is that you have taken as per your query only? Yes, Mr. Mahesh?
Ma'am, I was taken 2 sequence from hexokinase and 2 sequence from glucokinase.
Very good. So what is your output?
I got like this, ma'am.
Okay, down you go. No sequence conserved regions
Yes,
are
ma'am.
not there, even in one region. Oh, that's interesting. Okay, like okay, now you have taken
Now we got well. Now I got.
down you got. Good, good. So you can see that as you go down below, you're able to see some of the sequence. Some regions are only N N N, that means it's only nucleotides. Okay, fine. What about your guide tree and your phylogenetic tree? Is it telling what is the organism you have taken for exokinase and for the other one?
One is Homo sapien, ma'am.
Okay. Okay, let that we will see later. So hope you are now you are able to experiment yourself, right? So you had a query, he has just given the data, retrieved the data from the data set and he has experimented and you can see is able, Jay Mahesh sir is able to run the tool and get the output. That's very nice. Very good. Okay. Highly
Can we stop
appreciable.
it, ma'am?
Yeah. Thank you. Thank you. Anybody else? Yes, anybody else? You should come out of your shells. This is an open platform. Yes, people should be active enough.
Ma'am, I just did for the cry protein as you did.
Yeah. What happened?
So can I share it?
Yeah, please share it once. Okay, Manavnya is sharing the screen. Okay, what is that protein you have taken for your comparison or sequence similarity check?
It's a cry protein, ma'am.
Acha, protein only have taken. Fine. So you can see there are 1 or 2 regions, conserved amino acid regions, and some you can find there is some similarity among the amino acids. Very nice. Okay. So you're able to run it very easily. Fine. Okay. Very good. Nice. So you can, children, when whoever is, or the participants, whoever is running or using the tool, what you can do, my advice is take the screenshot, okay, of all the work what you're doing and paste it and keep it somewhere for your future purpose. Okay, hope you understood what I'm talking. Inverted commas assignment. Okay. So hope this is okay. So others might be you're still working on the tool. I will show you one site, what I told you that is called as Anusha. Yes. Then I will show you a tool that is open source that is called as Galaxy. If anybody is interested, I'll show you how it will run. It's very easy to utilize, use that one. So you have to register and you have to log in. You'll get a mail to your mail ID when you're registering, and you have to activate the link. Once you activate
What
the
is
link,
Galaxy?
I will show it now. I'll show it now. I'll show it.
Excuse me, ma'am.
Pardon?
Ma'am, I have a doubt. Ma'am, I was
Yes.
trying to run the code on Clustal Omega, and I tried to run the code by putting an accession number, but it wasn't working.
You tell me the accession number from where you have taken.
Ma'am, I've taken it from the FASTA by converting the data into FASTA, and then the first code that is given, I just copy pasted that, but it wasn't working.
Did you take the accession number for a nucleotide or a protein sequence?
For a protein sequence, ma'am.
Did you select in the job running profile as protein?
I did.
You did. How many accession numbers you gave?
3, ma'am.
3 you are given?
Yes.
Yeah, okay. So what you do is that did you take it from NCBI or from which database center you've taken?
NCBI, ma'am.
NCBI. So it is for cry protein or some other protein?
Cry protein.
Cry protein. Okay. So maybe you refresh the ClustalW page and again in NCBI take the accession number. Every accession number may have .1 also with them. So make sure that you're completely copy pasting only the accession number without anything, and then you try to paste it and check it out whether it is able to run. One more time you check it out. Okay? Yeah. So I will show you this one also. This is Galaxy. You have to register. So initially I will show it to you. Maybe then you can, so go to Google engine and then type Galaxy. So this is galaxy.org. So since I have registered, maybe it must be opening directly. So this is what is a tool which you can also use for running certain programs in this one. Now maybe I will share the URL to you in the chat box. You can also try open this link. Just check out whether the link is getting opened up so that you can register. Okay, I shared the screen. So what we will do is that first let us try to understand this Galaxy. It's all the details are given. It's an open source web-based platform for data intensive biomedical research. You can start directly if you are a member or if you have registered for Galaxy, or you can take the, they'll show you how to use the tool over here. Okay, so if I was interested to know who created, what is it all about. So you can see that the team is a Galaxy team, which is a part of Center of Comparative Genomics and Bioinformatics at Penn State, the Department of Biology at Johns Hopkins University. So if you just go below, you can have all the other data what you want to know about the Galaxy. So it's an open source which you can also use as a tool to retrieve certain sequences, analyze the data, and get the output over here. Okay, so now you have 4 different sections of your Galaxy. Now, first coming to my right hand side, you check out. I'm just showing over here. You have one first component which is called as the upload. So you can download and upload the sequence, because when you want to work with your Galaxy, you have to retrieve the sequences and upload over here, and then you have to work with that one. So I'll show you how to do all those things. Then this is a tool. So there are a lot of tools which you can use. For example, if I click this one, it will ask me to upload the data. So I'll show you that one also now itself. So what I'll do is I have to choose the local file. Since we are working with multiple sequence alignment, you remember from NCBI, I have downloaded what sequence in the FASTA format. So I'm uploading the same thing over here also. So once I have uploaded, it is just all are in color, and here you can see it is only 0%. So you should give the word start. So what it will do is all the data what I've downloaded and saved in my downloads, it has been now uploaded into the Galaxy platform. Okay, now I will close it off. So now I will be having the data. You can see once initially it was gray in color. I did not use that particular part in the Galaxy, but once I opened it and once I have uploaded the sequence, now it is becoming green in color. So when you are not using the tool, the whole platform will be gray or ashen color. Once when you upload, when you do some activity, and if that activity is completed 100% positive, then it will become green in color. And if it is running, it will be still in ash in color. And if there is any error, okay, no ash, no green, it will become red or pink in color. Okay, now you see the difference in the tool what I've shown you. At one left hand side, upload has become green in color. Similarly, to my right hand side, you see here, are you able to see here the sequence what I have uploaded? The greens is coming out, which is the sequence in the FASTA format what I have uploaded here. So here you have a view, that's a symbol for viewing, displaying your sequence. Then this is for editing in case if you want to edit any of the attributes, and this is for the deleting or discarding the content from your history, where whatever you're working in the form of history, it will be observed over here. Okay. So now you understood the link, how the tool will be working on. So now the next one is the tool. So if I click here, the tool, you can see so many list of tools are there. Get data, send the data. Then you have join, subtract, group, or FASTA queue is there. I'm just showing you. You can check out. You can once you have registered and logged in, you will be able to do it. Now we are going to work with which one? Multiple sequence alignment. So I have selected multiple sequence. Under that, so many tools are available, but since we wanted to do with ClustalW, we can use ClustalW only here. Okay. Now, once I click ClustalW, what you see in the center workspace, you can see the tool getting opened up over here, ClustalW, multiple sequence alignment program for DNA or the protein. And another parameter you see here, tool parameters are there, where you can see it is in the FASTA file. Since I've uploaded earlier and I've selected this tool as ClustalW, you see here what has happened. The sequence that is in the FASTA format has been already available over here. So it is already been uploaded into the Clustal Omega. Okay. Then if you go below, there are so many other parameters. If you want to select and run the tool, you can. Otherwise, you can just go and say run. Okay. So now you see what is happening to my right hand side. You see the tool 5 is your sequence. You see now it is in color has become, initially it was gray, now it has become pink. It's trying to run, the tool is run. You can see within few seconds I got the output because it has completely changed to green. Okay, so now let me see this one, first one, which you can see this is your tree, that is a guide tree what you are getting over here. And going to the next one, ClustalW, you should be able to see the aligned sequences. So it has aligned, and same thing, asterisk, colon, or it will be showing you the or the dot, whatever we have interpreted, what is the result you will be getting, you will be able to get it out. Okay, so this is how you can use a Galaxy tool. So this is another platform where you'll be able to do it. So for this thing I showed, and if you go down below more, you can see protein modeling is a, genome editing is a, filtering of any sequence contamination, statistics, machine learning. So you can see all the various tools. You have NCBI Blast Plus. You can just look into all these tools which are available with your Galaxy platform. Okay, and workflows are there in case if you design a workflow for certain programs to be run over here, you can take it and upload, or you can work with this one. And visualization of the results in different formats, all the things again, heat map, bar graphs, because sometimes some of your outputs, if you want to view them here, you can take it for visualizations. Whatever must be the way in which you want to view them, you can select it and have a look at it. And this is history. Show all histories it's telling. Since I've been working with that one, it is showing you some of the histories what I've been doing with the Galaxy earlier. Okay, multiple view. So same thing, it's showing me all the work which I've been doing it earlier also. That's history, how we have been working with that one. Okay. So that is how the Galaxy tool or the platform will be working. And as I told, if you want to edit, you can edit, or I can just go and say delete. It will be getting deleted from my history. Okay, so you can use even this Galaxy platform also to check out and get your output on the results. Okay, so I have given the link for Galaxy in the chat box. If you just click it, first you should register, and then you should be activate the link. So give a correct proper mail ID, activate the link. Once you are activating it, then you'll be able to access the Galaxy. So maybe some of the whatever is possible to show, the Galaxy is like a platform where you require very strong internet, as well as time is required, because when we do certain type of analysis, it will take some time. That's it. So because only 3 sequences we have downloaded and we have checked with the Galaxy. If you have registered and if you are able to access that one, then you can NCBI I told you can select many number of sequences, download in the form of a FASTA format, and you can upload all the sequences in your Galaxy platform. Just wait for 1 or 2 minutes, maybe you will be able to get the output. Now, I just showed you without selecting any parameters. Now you can play with that one, right? So what you do, first you do with pairwise alignment, that is a dynamic programming. The tool is there in the Galaxy. If you are checking the Galaxy when I showed you, the first one what they give is dynamic programming, which is pairwise alignment. So it will be giving you the output. So what parameter you select over there, based on that one, the output will be shown to you. So you can just work with that one also. You should get used with all these things, right? So maybe it will be an add-on, one more tool what you will be using in basic small study purposes. You can try and tell I have worked on with Galaxy, and this is output what I've got from that one. And you can compare also. Okay, from your original EMBL site you have taken it opened without, might be that is my okay. Yeah, you can use, that's what I'm telling. You can you can take the data set and you can try it out. Of course, you can try, but the only thing is that if more data set when we give, it will take some time. So you have to wait patiently for the output to come from that one. So first, initially to learn with it, you just try with the small data sets, and you can understand. And now I told you to play with the parameters. What do you mean by the play with the parameters? You first do with pairwise, then you go with multiple sequence, then you other parameters also you try to select it and compare. Now when I take only two pairwise, what is the output? When I take multiple sequences, what is the output? When I take the other parameter ClustalW with character counts or just only ClustalW, what is the output? Is there any differences or it's giving me the same result? This is how we experiment ourselves to learn more regarding what we wanted to learn, right? So you can try it out and experiment it. So we try to show you what are the sources from where you can access it and what to do. Further, we have to experiment and check it out. So that you can try it out. Of course, Bushra Khan ma'am, whoever it is, we will be sharing all these things, but not day-to-day basis, because after the sessions are over, we will be downloading the recordings and saving it day-wise. Once it is over on the last day, we will share you the link with the permission from the HOD ma'am, and then we'll be posting it to you. PPTs or whatever, we have to share it from our side with official information and the permissions. We will be doing it accordingly, ma'am. Okay, so I have done from my side. Yeah, thank you. So I have done from my side today's teaching process. Now, time for me to maybe to ask some questions, and you are supposed to answer. Or if you have any queries, you can just put in the chat box. And if anybody is still struggling with something, maybe we can help you out. Maybe Padma ma'am can help out with this question. MSA can be done with R programming or Python. Padma ma'am.
Yes, Vanitha.
One question has been asked by a participant. Can we do multiple sequence alignment with R programming or Python?
Yeah, you can do multiple sequence alignment using R program and Python. When we are going at the end of this course, anyway, we are going through basic BioPython tools and R programming. Maybe that time you can check out, and if you are already familiar with it, you can go ahead. Otherwise, we will be doing it in that session. We will see how best we can do it using the BioPython or R program. You can do many things using them. All these, what you are learning, retrieving the sequence, doing a sequence alignment, all this also, there are codes which can be used in BioPython and also in the programs. Our program has different tools. We can use all those tools to analyze, complete the genome data, starting from retrieving the sequence, doing a pairwise alignment, doing a blast, doing multiple sequence alignment. Anything related to genomics and proteomics, you can do with BioPython or R programming.
Okay, and Anvita, it's highly appreciable that you wanted us to send the recordings day by day. Okay, we will have a discussion with the HOD ma'am, and positively we will try to give a positive response to your query. We will try to help you out, not an issue. Okay, we will do that one. We'll discuss with HOD ma'am and we'll get back to you. Okay, so see, there are two terminologies. Whenever I teach for students, I say that there are two terminologies which I used to tell. One is clear, the other one is all clear. Clear in the sense, what I have told you, you have taken it, you have received it, and you have stored it in your brain. When I say all clear, it means that when I taught, it has gone here, and after some time from here, it has gone there also, outside. It has gone outside. So maybe if it is clear, maybe I expect one capital C in the chat box. So how many C? Okay, I get two C's. Okay, capital C's are coming out, working out. Maybe you have kept your caps. Okay. Okay, so it's clear. Fine. Okay, okay. Thank you so much. Do you have any doubts? If it is doubts are there, capital small d. If no doubts, it is small and small d. Nandini Sharma, what is AB? Anirban, you are saying N? ND it is. Fine. So I hope the first 5 minutes what I told you has been working out very well, that open your eyes wide enough to see what we are telling you and keep your ears wide enough to listen what we are going to tell. And I'm so happy that you have been along with me till the last and no disturbance, and it has been a very good session. So happy with all the participants, and thank you so much for your cooperation. And one thing I would like to say is that every day we'll be posting the attendance in the form of Google form to you in the chat box. And we expect you to please respond it. Just your name and your college name may be asked over there, nothing else. Just 5 seconds or 10 seconds only it will take. Please do that one. This is to make sure that whoever is attending is being made a note of that one. Okay. So any queries if you want, you can ask. Padma ma'am, anything you want to tell? Or shall I send the attendance sheet or shall we wait? Anybody is having
Anyone
any
has
issue?
worked out using Galaxy, you have been successful in logging in and you have been able to do it. You are ready to share it and show whatever you are able to do using the Galaxy. If you have any initial teething problems, where to look for that or where to click or anything, we will guide you through if you are able to register through the Galaxy now. Since we
Yeah, we
have
have
still
some time, right?
left.
So you can, sufficient time is there.
And another point I wanted to add regarding the ClustalW sequences, whenever you are trying to take it directly from the NCBI database, you are looking at the sequences. Make sure that you are looking at diverse sequences. That is, you are taking from different species, not just from one particular species. And also, the number of amino acids which are there in the samples what you are selecting or with the accession numbers. Somebody has shared their screen. I was observing that out of the 3 sequences what you have done, only 2 of them or 1 of them there is a sequence throughout. Other 2 sequences there is lot of gap. So what would happen in that case is you will not be able to get the actual significant sequences which are there amongst the 3 sets of samples what you have done. So if for that means what I mean to say is, if you are taking a sequence of length 400 amino acids, another sequence of 200 amino acids and another of 600 amino acids, the alignment would be done only for the best matching set, and the rest of the sequences would not be able to be matched there. So it is if you are learning it for the first time and you want to see the impact of that tool which you are using for learning the alignment of sequences, preferably please pick up sequences which are distant organisms of different species, from like diverse species, and also amino acid length, number of amino acids. It will be given there when you go into the NCBI and look into the protein. It will tell you how many amino acids are there, 403 or 420. So near about that you can take, and you make sure that you take just not 3 sequences, at least you should be having 7 or 8 sequences, in which case, and when you are going to do the evolutionary relationship, you will be getting a very clear picture which 2 species or which 2 sequences will be closely related to each other.
Yes, hope you got what ma'am said. So when you're taking the sequences also, make sure. Anusha, you had a trouble initially. Maybe you were able to sort it out, or still you're having some issue?
Yes, ma'am, I got it.
Got it. Okay. Ma'am, I've tried to take insulin, but when I have used gene insulin, I'm not able to convert the file to FASTA. But for protein insulin, can I know why?
You can
Mercy
share
Raj.
your screen and show your problem. Then we will
Problems.
be.
Yeah, we'll be able to sort it out. Mercy Rajkumari, whoever it is. If you
You
can
can
do
share.
that one,
Yeah, please
we
share your
can.
screen and show me what the problem you are facing. Please share your screen.
Because we don't know where the problem is, right? So if you have taken gene insulin, you have typed in the NCBI as, yeah, she's sharing now. So you have taken the gene as insulin. Okay.
But here you are showing insulin receptor, insulin like growth factor. There is no insulin there, right? So insulin is different, insulin substrate is different, insulin like growth
Insulin
factor is
receptors.
different. All these are different. Insulin protein, yes, it is coming, but you need to search down, go further where there is only insulin, not insulin like gene factor or insulin like growth factor and all that.
So when you are doing all these searches, no, when you are downloading for a gene or for insulin, like what the problem has arisen, you be very crisp in trying to take, okay, gene, I want to take for insulin. When you want to be taking only for that one, don't go for that. Many number of data will come out, right? So you select only gene, insulin, Homo sapiens, gene, insulin, ratus, or that is a rat, or for a mouse. Like that, you go specifically only the gene related to your query, that is the insulin. Now you again try it out, Mercy, and see whether you are able to sort it out. Don't take receptors and all. You can just try it out that one and check it out. You will be able to get the output. So others, you don't have any issues. Everything is fine for you. Galaxy, anybody has tried? Anybody has logged in and registered? Were you able to do that one? Yes, ma'am. So can you share your screen and tell to us?
Yes, ma'am. One more.
Yeah, please. Others also will know, right? Did you run the tool or only you have till now registered? And
Both, ma'am. I have registered and run the tool.
oh, that's very nice. Good. You have to be very fast enough. Others, it's a learning process. You have to learn quickly. So that is very nice. Okay, you have taken the sequence, downloaded. Oh, what is the tool output you have taken? What is the parameters you have selected? That's very important, right?
Yes, ma'am. That is the visualization I have done for the multiple sequence alignment.
Yeah, very nice. Okay, okay, good. Show me that tool which you have used for visualization. You have separately selected the tool visualization and you have checked it out.
Yes, ma'am.
Very
In
nice. Good.
visualization, I have multiple sequence alignment.
Okay. Very nice. Okay. So what is the protein you have used for your Galaxy tool?
Hemoglobin, ma'am.
Hemoglobin. Okay. So you selected the hemoglobin from NCBI and you have downloaded the data set and then you have uploaded in the Galaxy.
Yes, ma'am.
Okay.
Yes, sir. Just to go back to that NCBI one, there you have taken hemoglobin protease, right? Not hemoglobin.
Yes, ma'am.
Yeah. So all other sequences are only protease. You are comparing a protease with hemoglobin, or what is it? Because here, whatever you have selected is all hemoglobin only. 3 of them are hemoglobin, but the first one was hemoglobin protease. In the first one,
Two are hemoglobin proteases and two are type 1 and type 2 hemoglobin partial from another species. I mean, from another organism.
No, the protease is different from the protein, right?
hemoglobin.
Yes, ma'am.
So, yeah, That is from microorganism, E. coli.
Yes, ma'am.
So yeah, so how many amino acids are the see here the number of amino acids are around 1376, and
Yes, ma'am.
the other section, other amino acids, what are the number of amino acids?
1765 and
Okay.
2148 and 1377.
Okay.
Okay, that's good. That's very nice.
So what is the difference you observe between hemoglobin and hemoglobin protease? Can you find out the difference from your alignment?
With the accession number, you check out which is protease and which is hemoglobin and try to give us the output.
Okay, ma'am.
Okay, anybody else? Anybody has tried with the Galaxy? You are registered now and you have used the tool, huh?
Yes, ma'am.
Okay, that's nice. But as ma'am is suggesting, you be very crisp in your search, participants. Like wide thought is okay, but when you are interpreting the output, they'll ask you with what you have compared with. Means we cannot tell different sequences or sizes or whatever it is. You are now this is only to we have taught you, but now you have to be very careful when you are selecting the sequence, when selecting the species, what is the size of the sequence, and then giving the input sequence for your search process. Okay. Ma'am, shall I post the attendance sheet to them for day 2?
Yes, Vanitha. So I thank Dr. Vanitha for giving a very wonderful explanation about cluster alignment and using the different tools, how to align the sequences, how to analyze and how to interpret the results. I hope the students will take it forward and practice the tools which have been shown today in this session. And we will be continuing to use this alignment again when we are going to do the phylogenetic alignment and the subsequent. There will be one more session which will be coming up about pairwise sequence alignment later sometime. So in that case, you will be able to appreciate the difference between pairwise alignment and the multiple sequence alignment. Okay, then you can all complete your attendance and we can wind off for today.
Any queries are there? If you want to ask, I'll stay back. Others, after giving attendance, we can wind off the session for today. And thank you, Padma ma'am, for giving me this opportunity. And thank you all the participants for good cooperation. And thank you so much. So if anybody is having any query, you can stay back and ask me. And otherwise, you can finish giving the attendance. Assignment, we are not giving day wise. What we have decided is 4 resource persons are there, right? So each one resource persons will be taking 3 or 4 topics. So once when all the topics are over, we will be giving you the assignment. That's what we have thought about it. So today only is my first day, right? So I have some more sessions to be completed. After that, we will give you the assignment details. Whatever you have been working only will be your assignment, so you don't have to worry about it much. Be regular. That is more than enough for us. Reading your question, Srinidhi, if in case we want to do a ClustalW analysis for different plants of the same species, like Solanum, for example, by what aspect can we do the comparison, ma'am? So under Solanum, you have 3 or 4 species, right? So that is what your query is. So in that case, you can take up protein or whatever particular gene of interest if you want to take. Check out that one, and you can compare and check it out whether now when the genus is the same, when the species are changing for that particular gene or a nucleotide or your RNA or your protein, they are different or there is any conserved regions among them, you can check it out. Maybe you can work it out and check it out. And from the database, wherever you are searching for it and trying to download, you search only for Solanum and that particular gene or protein of your interest in that particular Solanum genus. If the data is available, then you can compare it and check it out. Because genus is same, right? If that is the query and if I've answered correct, it's okay. In case if still query is there, you can just type again. Srinidhi. Okay, thank you, Srinidhi. Anybody else? Anything else, or shall I wind it off?